Well, welcome everyone. I'm so excited to have everyone here. I wish I could see everyone's faces, but I'm Erin Johnson. I'm the Director of Development for Neurosciences here at the UNC Health Foundation. And I'm honored to bring to you Dr. Todd Cohen, who is our leading researcher and expert in memory disorders at UNC and really in this whole space of North Carolina. So we're really honored to have him here at UNC, and he's just a treasure to all of us and to the future of helping memory disorders. So I thought I would kick off with just a few tidbits about Todd and about how this webinar is going to work, and then we'll get right into it and we'll take Q&A throughout the whole event too. So a little background on Dr. Cohen. So I'm very, very lucky to get to work with him every single day. He is one in a million, and we're just really grateful to have his whole sense and interest and excitement about this space, and is really necessary in this age and time. So Todd grew up in Philadelphia and Pennsylvania area. He went to Penn State undergrad. He then went to Fox Chase Cancer Center. So he thought he was going to go into cancer. And then he went into Duke for his PhD for molecular biology. He did not have a plan to really get into memory disorders necessarily, but his grandmother got sick. And so he ended up at a postdoc training at UPenn in Alzheimer's, which is what his grandmother was dealing with. He got hooked. The interest was there. He was ready to pursue this as a career path, and he ended up coming and here and starting his lab at UNC in 2014, and he set up a brand new program in Alzheimer's disease. So we are the leading program in the state right now. Really honored to have him, and just please ask questions. He's an expert in all of this. But one thing about the questions, so is a Q&A feature. I think you just saw that on the slide before, but you can go in there and ask questions. We're going to take some breaks in between some of his slides and take those questions, and I'll help ask them of him. And then I'm going to kick it off to Todd by just saying this is a really hard time for everyone. Webinars, everyone's a little bit zoomed out, so we're really grateful to have you here, and especially during a week of this thing called Gratitude Heals. So this is a week of gratitude because we're really grateful for all of you who are giving back to the UNC Health Foundation, who are making the research, the clinical work, and all of the important things that UNC Health is doing right now possible. And without your all support, I don't think we'd be able to do half of this. So very, very grateful to have you. And please ask the questions. And Todd, if you don't mind kind of telling us about how you are surviving at COVID, how you're doing through this weird pandemic, and how your lab is surviving as well.
Yeah, of course. Thank you, Erin. Just want to say thanks to Erin for the introduction and for Allison behind the scenes here, who put this together. And I also just want to thank all of you listening. I know your support, thank you for your support and for your dedication to UNC, because we certainly need it, especially over the last year or so. Yes, but as far as me personally, the last year has been challenging. And I think, like everyone out there, we're dealing with homeschooling, and I happen to have two girls that are right in that age that is difficult for 6 and 10 years old, which is exactly the ages where if they were any younger, it might be a blip in their memory, and if they were any older, they'd be able to deal with it. So we're right in that sweet spot of it being challenging. And so combine that with running a lab and trying to deal with the challenges that COVID has presented has been a very difficult year. But I am fortunate to be surrounded by great people like you, Erin, and like everyone who's committed to helping to solve some of these horrible brain diseases. And I know that many of you listening, I know that I have not met many of you, but I appreciate all your comments, and I will do my best to respond to every and all comment, whether we get to it today or not. Okay, so please let me know what you're thinking. I appreciate all your thoughts. So I guess we'll get into it. How does that sound, Erin? I'll go ahead and share my screen. And okay. How does that look? Can you see that okay?
We can. I think you can enlarge it.
Okay. Okay. Um, so I thought I would just talk a bit about our work in the lab, even though it's taken a hit and we've had a difficult year. I have to say, I have a lot of very motivated and determined people in the lab to move things forward. And I'm very grateful to them. And we've actually made significant progress. So I think I'm more proud than anything in my career. I'm probably most proud of the progress we've made over the last year. And I'm going to try to tell you a little bit about the discoveries that we've made as we go. I won't fill in all the details just in the interest of time, but I will touch on all the new excitement that's happening in the lab. And I just want to show you in the new age of Zoom photos, you can see our group down here, and I'm just going to go to a pointer. You can see our group down here. There are around 10 of us in the lab with another 7 undergraduates. And so we have quite a large group. And so it's been difficult to accommodate a large group with the 50% capacity rules that COVID has instated on us. But nonetheless, we continue on. And if anyone is around here, the 6th floor of the Neuroscience Research Building on campus, please come say hello. Okay, so I just want to give you a sense of our goals and what we're passionate about. And so I've got a list here that I continually modify as I move on in my career. But we feel like there's 4 major things that we think about a lot. One is risk factors. And so the first point here is why some of us get dementia and others don't. And this is a major question because we want to be able to highlight that, that single or those few people in a population that might be on track to get disease. We also want to know what those positive risk factors might be, the things that can protect against disease. So a lot of the talk today is actually going to be focused on risk factors. The second thing we care about a lot is genetics. How do your genes determine whether we get dementia? And in addition, which genes can actually prevent you from getting dementia? And so one of the things that I'm very excited about is a new gene therapy approach that we've developed in the lab where we hope one day we can deliver the genes that we find back into an individual to protect them. So number 2 is genetics. Number 3 is this concept of aging and what aging even means. And after all these years of thinking about aging, I've come to the conclusion that everyone views the word aging in a slightly different manner. But at the end of the day, there's something about age, your clock, your normal body clock that is linked to dementia. In fact, it's the biggest risk for dementia because children, for example, don't get Alzheimer's disease. This happens in a context of an aged brain. So some of what I'll tell you is related to how aging is a primary driver of dementia. And then I think what everybody here cares about very dearly is, can we develop stuff? And I use the word stuff rather than some other technical jargon because I really mean stuff. I mean materials. And those materials, whatever they are, we hope they can be used as a therapy. Now, for example, this could be a drug, a pill, but it could also be a virus, a helpful virus, not a harmful virus, as in coronavirus, but a helpful virus that's showing promise in the clinic. There's also techniques. You could imagine deep brain stimulation and other techniques, changes in light dark cycle, for example, that might actually be therapeutic. So we really want to use 1, 2, and 3 to help inform our development of things that can beat this disease. Here's why we care about these diseases. Now, this is just Alzheimer's disease, but I just want everyone here listening to know that dementia is not just Alzheimer's disease. That's just one form of Alzheimer's disease. In fact, it's the most common, but there are lots of types of dementia. It happens to be that Alzheimer's disease is increasing at alarming rates. And if you go out here to 2050, I think you can see in the US alone, we're estimated to have over 12 million cases of Alzheimer's disease. And so I'm thinking about this trajectory being very difficult to manage, both medically. We need doctors, we need the right support system to tend to these patients. And so I don't think we're going to be equipped to deal with this. And I just want to highlight in red is the sort of the elderly population, while in blue and in green are the sort of the younger ages. And so you can see they're all increasing, but the incidence of Alzheimer's is going up at an alarming rate in this more elderly population. And so we need to figure out ways to prevent this because it will come back to get us if we don't. We don't want to be ill prepared for this. And so I just wanted to sneak this slide in here at the beginning because I know all of you listening probably have friends, relatives, etc., that have that may have been affected by a form of dementia. And I put in this slide just to let you know that they're all very much, or many of them are very much related. And so Parkinson's disease, Lewy body dementia, Alzheimer's disease, not many people realize this. There's a common misconception that these are separate diseases, when in fact, half of all Alzheimer's patients have brains that look like they have signs of Parkinson's disease. And so I think this is really helpful for us to view not every disease as an individual one-off disease, but rather as a spectrum of diseases. And this graph on the bottom illustrates this in another way by comparing frontotemporal dementia, which is a type of dementia that it affects behavior, which is very similar to Alzheimer's disease, but they're not the same. Moreover, another syndrome called ALS, which is thought to be a motor neuron, a motor disease that affects movements, is actually also highly related to frontotemporal dementia. Many patients with ALS or Lou Gehrig's disease end up showing signs of dementia. And so it's very important for everyone here to know that these diseases operate on a spectrum and that someone can have mixed elements of multiple diseases. And so we need to find commonalities so that we can treat not just one, but hopefully all of them. Okay, and so risk factors. And so a risk factor is a factor that changes the course of disease. And so here's a baby on the left, and then you have a grown man, an elderly man all the way on the right. And so very long time ago, maybe 10 years ago, I wondered why children don't get Alzheimer's disease. There must be risks that put someone, an aged brain, at risk for Alzheimer's disease. And so when I use the word risk factor, I mean some element, whether it's your genes, whether it's age, whether it's environment, that causes the brain to change in a pathological way so that it's more susceptible to disease. And so here's some examples of risk factors. The ones in red are the ones that our lab has very active programs on to try to understand. And the ones on the left, age, female gender, ApoE, other genes, diabetes, and head injury. All of these factors are very well known in the literature and in the field to be very, very likely, if not definitive, factors that can put you at risk for dementia. Okay, now we're very interested in age and very interested in genetics, but there's a lot of literature on head injuries, for example, football players and boxers who, by virtue of being concussed, are at higher risk of dementia. Okay, now the risk factors on the right in the green box are likely to be involved, but there's quite a bit of controversy surrounding these factors. And I know several of you listening may have posted questions or are interested in some of these. And I'm going to address some of them in red, but suffice it to say, there is still controversy, for example, in whether viruses can cause Alzheimer's disease or dementia. Pollution and exposure to toxins or diet also they're gaining traction. but there's less solid evidence for this group on the right compared to on the left, okay? And I'll touch on several of these in the next few slides. The one that we're very interested in there at the bottom with the stars is sleep. And all I can say is that we should all be getting our 7 to 9 hours because it certainly looks like sleep is gaining as a positive risk. In other words, getting those hours of sleep can really protect against impaired memory. And I'll talk a little bit about that as well. Okay, so here's our trajectory as humans, right? We start out as babies. We go through this adolescent period, teen period, adult period. And so I have that highlighted here. And so somehow when we are in our, and this is just a rough estimate, but I want to get everyone on the same page. Somehow when we're in our 30s, 40s, 50s, changes happen in the brain. And by looking at brain tissue of many, many patients, it looks like when we're in that middle-aged period, things accumulate in the brain. And in this case, we're seeing a picture of plaques. Plaques are these deposits of protein called A beta that appear to start accumulating. Now, this is rough, but say in your 40s or 50s. And then what happens is there's a gap period. This period could be 30 years, for example. It could be 20 years. It's variable from person to person, depending on lots of factors. But then what happens is inevitably, if a person gets late onset Alzheimer's disease, they get a brain that is full of these tangle proteins. And the protein that makes up the tangle is called the tau protein. And it appears that this tau protein is the key protein that is driving a lot of the neurons to die. And so we spend a lot of effort trying to prevent this from happening. And essentially, when your brain starts to look like this, you are on a trajectory to show significant memory impairments. Okay, and so in every Alzheimer's patient that is showing memory deficits, their brain has some extent of tau tangles. And so the current thinking is that if we get rid of these structures at this stage, this could be beneficial, okay? And so we're very interested in what's happening from this period onward. And so I am approaching my mid 40s. And so this gap period is a critical period where things are happening in the brain. And I really want to know what those things are, because I feel like if we get it at this stage, we can beat this disease, okay? So earlier this year, we published a paper, which we're very proud of. And so especially in light of COVID, we call this a COVID paper, not because its topic is COVID, but because it happened during COVID. And it's remarkable to me that this was even doable. I just have a super motivated and determined bunch of people in the lab. But here's the title of the paper. And to avoid all the technical jargon, we can translate this title into this. Tau clumps are taken to waste containers in the brain during aging, but something goes wrong and they linger. So that's what we found. And we think that this could explain what happens in that intermediate gap period when someone's aging, but doesn't quite have full-blown disease quite yet with symptoms. And so here's what we found. And I like this picture because it's worth, they say picture's worth 1,000 words. Essentially, what we found is that there is a very angry cell called an astrocyte that is present in everyone's brain. But as you age, this astrocyte tends to get, we're saying irritated, but this astrocyte tends to get sort of inflamed, and it starts to do damage to the brain. And so what we see here is in red is this cell with its arms. Each branch you're seeing here is one extension of this astrocyte. And what it's doing is it's grabbing on, and you can see it maybe a little clearly here, it's grabbing on to these green blobs of tau, this protein that's accumulating in middle-aged mice. These are mice, these are not humans. And it's trying to get rid of it. It knows there's a problem. It's seeing a very early type of this tau protein, and it's saying, hey, we better do something. We better act on this and get rid of it. And so the remarkable thing about this is that we can image this now, which is, a tribute to technology. The other remarkable thing is that this is in a 1-year-old mouse brain. Now, this 1-year-old mouse is somewhat in its prime. It's the equivalent to about a 40-year-old human. So it's not going to show any signs of brain diminution for quite a while. And we're already seeing this in essentially middle age. So what does this mean? And I just want to actually back up and acknowledge Connor, who is a grad student in the lab, who did a lot of the work on this project. And it was just recently published. So please feel free to check the publication and reach out to me with any questions about that. But so what does this mean? Well, we think what it means is that there's this critical gap period as we age where your brain is fighting this burden of protein that's starting to accumulate. And if we let this go unchecked, eventually 20, 30, maybe even 40 years later, we start to surpass what your brain's able to tolerate. And that's when memory issues start to begin. So because we've made this discovery, we're now following up on this and saying, well, can we detect this in live people? Can we see this in people and actually show that this is real, not in a mouse, but in a person? And so what we're doing is we are taking blood and spinal fluid from people as they age, just normal aged people, people that may be on course to get Alzheimer's disease, but people that may not. And we're storing their fluids and we're analyzing them in very sophisticated machines. On the one hand, we have a mass spectrometer, and on the other hand, we have a plate reader. Now, these machines allow us to detect proteins of interest. They're very sensitive and very specific. And so we're asking these questions here. Do middle-aged people produce these tau clumps in the brain? Normal aged people, my age, essentially. I'm 42. Do people in their 40s produce this tau? Can we use this as a biomarker to diagnose dementia that's not present, but that may happen soon? And can we distinguish among different types of dementia? And so remember, I told you there's this umbrella of dementias. Can we say that one person might have Alzheimer's, but another person might have frontotemporal dementia, for example? Now, why would that be beneficial? Well, it could be that medications are more appropriate for an FTD patient versus an Alzheimer's patient. And in that case, we might want to have something that distinguishes the two. And because we're on track and moving forward, even during COVID, we should know the results within the next year and a half. And the reason is we've been able to assemble a multi-institutional collaboration to address this very question. And this involves many people at UNC, Duke, University of Pennsylvania, and University of Miami. And that just got initiated a month or two ago. So we're very excited at this finding translating both to a biomarker, but also to a potential therapy, because we're trying to block this form of tau from accumulating in the first place. And so we'll keep you posted. And Allison, if you'll have me back in a year, I hope I can give you all an update. Okay, so the next risk factor I want to talk about is sleep. And this will just be a little brief snippet. And I'm really excited to work with Dr. Graham Deering in the cell biology department, who happens to be a sleep expert. And if you all don't know the culture at UNC, it's a pretty special place. It's a place where I can go get a coffee and meet Graham at the cafe and have a conversation, and then all of a sudden we collaborate. And that sort of camaraderie and atmosphere at UNC is very rare. And I don't know if it's built into the culture at UNC or how it evolved, but it's a wonderful thing because it allows us to ask questions that wouldn't be possible with one person alone. And so Graham studies sleep, And we all know that sleep is very important. Everything sleep, all mammals sleep. And so here's a picture of a fly, a mouse, and a baby. We need sleep. It's essential. It promotes learning and memory. It promotes attentiveness. It controls emotion. Um, and so more recently, lack of sleep has been implicated in Alzheimer's disease and other dementia. And again, it's not clear how, but Graham and I have teamed up and we now have ongoing projects to understand what happens if you deprive just a little bit. What happens to your ability to learn and remember? What happens if you force sleep? Is that protective? And so we have a mouse house at UNC, an actual mouse house. And so there's a sign that says mouse house, and here's all these mice in their sleeping quarters, and they're being monitored, and their sleep is being carefully evaluated. And so we're able to look at how much they sleep and what their quality of sleep is. And so Graham and I have ongoing projects to look at what happens if we alter sleep. Does the onset or the progression of dementia change? What happens if we force good sleep? And you might be saying, how do you force good sleep? And take my word for it, Ambien and these other sleep promoting medications are not giving you the good quality sleep that you would get naturally. And so we've come up with new drugs that might be able to restore good quality sleep. And so we're now testing that in mice and asking what happens if we force good quality sleep? What happens to their learning and memory? You could imagine a therapy for dementia being good quality sleep. And that's what we're testing at the moment. So I just wanted to highlight this little snippet in the lab because I think it's going to end up being really high yielding and potentially produce new medications that are sleep promoting, and by virtue of promoting sleep, also memory promoting. Next risk factor, the environment. And so I'm sure that inevitably when I give talks or have sort of casual discussions, the question about our environment comes up and what things might be promoting dementia, in part because we're paranoid as humans to want to avoid those things. And I always discuss this type of a map when we talk about the environment because I'm always confused by it, quite honestly. This is a map of Alzheimer's incidence by state, and it takes all other factors into consideration, age, etc. And so this is a normalized map, which tells you that the highest incidence of Alzheimer's disease are in the states in red: Tennessee, South Carolina, Washington State, and the lowest incidence of Alzheimer's disease are in places like New York, Florida, Hawaii. There doesn't seem to be a lot of rhyme or reason here. For example, what does a New Yorker have in common with, someone from Nevada or Florida? And so what it boils down to is I think a lot of people think there are environmental influences that are controlling this type of a map. And so what could those influences be? Well, there have been several studies that have suggested that, and I crossed out noise here in the title of this article because noise is not a risk. They showed that it was a non-risk factor, but the pollution part appears to be a risk factor. And so exposure to elements in the air, particularly diesel fuel, has been shown to, within a certain radius, has been shown to increase incidence of dementia. And so if you see this paper, this is one of three really conclusive papers on this topic. Their conclusion was, I'll just read this to you: we have found evidence of a positive association between residential levels of air pollution across London and being diagnosed with dementia, which is unexplained by known confounding factors. And so there are toxins, there are pollutants, there are herbicides, there are fungicides, there are things that are out there that have some toxic properties, and unfortunately, the FDA doesn't regulate or control a lot of these, and so we have to be vigilant about our exposure. And so what we've done in the lab is we've decided to explore this a little bit. And I'm just showing a picture of Julia here rock climbing. Julia is a postdoc fellow in the lab, and she is embarking on a project to test the 1,000 most common pollutants, toxicants, diesel exhaust fumes, pesticides, herbicides as triggers for Alzheimer's and related diseases. And the way we do this is you're staring there at the bottom left here at a giant 384 well plate. And this well can be engineered to grow neurons in it. And so we can grow neurons in these wells and then treat those neurons with any one of 1,000 compounds that is potentially a risk factor. And Julia hopes that she's going to be able to uncover which of those pollutants is one that might be driving disease. And so I wanted this to be on everyone's radar, because I think this could really tell us which to avoid, whether they're present in food or drink, whether they're present in new homes that are being built, for example. We want to know this information, and I'm hopeful that now this can actually go very quickly. So we think that within the next year or two, we'll know which or how many of these compounds are toxic. And so again, I'll check back in with you once this study is further along. But we do have several compounds that look like they are causing toxicity. They just need to be replicated, and so we'll know that in the coming year. So I thought I would stop here and do a little brief question and answer break, and I'll maybe pose this one question, and then I can hand it off to Aaron. And Aaron, you can field some of the other questions, but I can start with one that I've essentially given myself here, which is, why study risk factors if we don't have drugs that are effective? And that's a common question, right? If many of the Alzheimer's drugs that we're currently given are not so effective, what's the point of studying all the things that cause the disease? And I would answer that by saying, clearly, you can't just study risk factors. You need to have therapies that are in the works that make the brain healthier in these patients that are at risk. And that's what I'll talk about in the sort of the second half of this talk. So that was my first question. And Aaron, why don't you go ahead with any other questions that we might have?
Sure thing. Thanks so much. So one question that came in was, what are the first signs of dementia?
Yeah, the first signs of dementia, unfortunately, once you're showing signs of dementia, that probably means that the disease is is further along than we would hope. And if you remember that timeline with the child to the elderly, dementia actually starts at the microscopic level. If you get it in your 70s, for example, probably in your 40s, in your late 40s, potentially your 50s. Once you show signs of it, that means you've lost quite a bit of the neurons that control things like your memory, your spatial memory, where did I leave my keys, things like that. And so it probably starts in your 30s and 40s, and it probably shows symptomatology when you're in a case of late onset, probably late 60s, 70s.
Thank you. That's daunting also.
Very.
Another question quickly, are there any supplements or foods that have been proven to reduce, prevent, or reverse dementia in any significant degree?
I would say to foods or supplements, it's all hearsay. There's nothing that's solid and proven. And I think the word was proven there. I would say that the answer is no to that. However, physical exercise, about 30 minutes a day, about 4 to 5 times a week, has been shown very repeatedly in many, many studies to be protective. And so if there was one positive risk factor, I would say, that was under our control, it would be exercise. But as far as vitamins, nutrients, any sort of dietary supplements, I think the data is still not quite there yet.
Good information. Everyone should bundle up and probably go for a walk today. I'm going to let you continue the presentation just to stay on time, and we'll take more questions in a little bit.
Okay. All right. And you can see this again, Erin. I think I got off for a second. Can you see that okay? Okay. Okay, hold on one second. It's not letting me. I'm just going to stop this for 1 second because it's not letting me, and I'm going to start it up again. Okay. Okay. So I'm just going to move on to sort of the second half of this talk and talk about what we can do and where we're headed with potential therapies. And I'm also really proud that this study came out over the last year, a study that we think will identify a potential therapeutic option to deliver a protective gene to the brains of certainly mice. And we're very hopeful that we can do this with people as well in clinical trials. And so this is a study that just came out last month from our group. And again, in lieu of the technical jargon in the title, I'm going to translate this to what you see at the bottom. This gene called HDAC6, which is called histone deacetylase 6, is protective and could be useful at preventing dementia.
Dr.
And
Cohen,
so, yes.
I apologize for interrupting. We can't see your screen. Do you mind resharing it?
Oh, I'm sorry. Let's see. How's that, Allison? Is that okay?
That's perfect. Thank you.
Okay, sorry about that. So this was a study that we just recently had accepted. And again, here's the journal and the article in case anyone is interested, and I'm happy to chat with anyone offline about this. But this gene that we found, that we discovered, called HDAC6, is protective, and we believe that it could be useful at preventing dementia by delivering this to the brain. And so our very challenging long-term goal in the lab is to take this gene and get it into the brain by a variety of methods and see if it protects those neurons. And so let me just take you through a little bit of this. So I hope you can all see this. There's a little video down there of a little white mouse swimming around a pool. And people don't always realize that you can do learning and memory in mice. And so when you want to identify whether a gene is protective or not, you can't just ask the mice questions. Rather, you test them in assays for learning and memory. This test is called a Morris water maze. And essentially what we do is we can take this mouse, we can drop it in a pool, and this mouse is very good at swimming around. And in one little area, there's a hidden platform. And this mouse knows that if he finds that hidden platform, he won't have to swim and won't drown, which is a very important survival skill. So this act of remembering where this platform is called spatial memory. And it's the same phenomenon that people use when they remember things in their house, for example. Which room is my bedroom? Which room is my kid's bedroom? And here you see a zoom up of this mouse. He's standing on a platform that's just below the surface. And I'm sure he's wishing that he had one of these at the moment. But unfortunately, we do not give them little yellow boats. They must find this platform on their own. And so sometimes it takes them a minute, sometimes it takes them 2 minutes. But the really smart ones remember where it is and find it very quickly. And so we can time how long it takes them to find that platform. And so this concept of spatial memory is very similar to what you would see in a home. And so here's an example, someone like me that can never find their keys and they're looking under the couch. And so where did I put those keys? I have 5 rooms. I know I walked in. And so that is that quality uses a part of the brain called the hippocampus. And the hippocampus is right here. It's this structure right around here. And that structure is what atrophies and becomes and essentially dies in end stage of dementia. And so that is typically what goes in patients with dementia, is this spatial memory. And if any of you know people with that have gone through the battle with dementia, some of you may be surprised to know that their long-term memory is preserved. And in fact, my grandmother could remember what she did in her teen years when she was in the very end stages, where she didn't even know which room she was sitting in, yet she knew what she did as a teenager and as a young adolescent. So a lot of it is because of the vulnerability of these neurons that control spatial memory. And so this is as sciencey as this talk will get today. And so I just want to give you an example of how important we think this finding is. So here's a normal mouse that has its HDAC gene intact. And when you ask this mouse, well, we don't ask them, unfortunately, we drop them in and we tell them to find this platform. He's really good, he or she is very good at finding this platform. So he runs around and inevitably he finds this platform. And so this mouse is protected from dementia. Now, if we get rid of this HDAC gene, these mice seem to show signs of accelerated Alzheimer's disease. And when we put this mouse in this pool and ask him to find that platform, he's not so good at it. We drop him in, he ends up in this quadrant, this one, he's in all the quadrants equally, and he's just not very good at it. He has not been able to learn this task. And so this is what this looks like. If I translate my words into a graph, what you can see is over time, the mouse with the HDAC gene gets really good and ends up finding the platform in around 10 seconds, a little more than 10 seconds, okay? The mice without this HDAC gene essentially just flutter about, and they're still not any better than they were when they started, okay? So this tells us that this gene, this HDAC gene, which goes down in patients that have Alzheimer's disease, we're losing that protective ability in the brain. And so what we now want to do is restore this gene back to patient, well, first to mice, but then back to patients that have this disease. And how are we going to do that? Well, here's an example of a person that has moderate dementia. This is what their brain might look like in terms of their load of tau tangles, okay? You could make the equivalent would be this poor mouse that has this accelerated form of disease. What we're doing in the lab now is designing drugs that boost their resiliency. And we think this gene HDAC6 is a gene that boosts resiliency. And so what we're doing is we're developing drugs, and we're also developing viruses, good viruses, not bad viruses, good viruses that contain this particular gene and other genes that I don't have a chance to tell you about today, to the brains of these animals and eventually to people as a way to make them better. And so we hope that by delivering these protective elements to the brain, we'll be able to ward off the signs of dementia, not just the signs of dementia, but ward off the symptoms of that memory loss that occurs as the disease progresses. All of this is ongoing. We've set up an entire gene therapy suite in our building, all dedicated to delivering genes to the brain. We think this will be a very, very effective way to boost resiliency. So this is what we're doing at UNC. I just want to let everyone know sort of the big picture idea, because you can't do these things in isolation. We're not a silo, and I think years ago, it may have been that way, where labs operated in a silo. Now we have to merge the clinic, of course, the people that see the patients, and this is made up of autopsy programs, clinical coordinators, and a whole slew of people that interact with patients. We'll merge this notion with imaging. For example, we can now image the presence of tangles and plaques in the brain. And UNC happens to be at the forefront of that field, an amazing center called the Brick Imaging Center, where we're able to use PET tracers to tell you in a live person whether you have the signs, the pathological signs of Alzheimer's disease. Now, whether you want to know that or not is maybe more of an ethical question, but we can currently do that as part of clinical studies. Now, imagine if you have that information, you have patient populations, and now you also have drugs and studies done in a preclinical model. When you put these things together, I really believe that magic can happen. And I think that's what's happening at UNC right now from the efforts of an awful lot of people. And I'll recognize a lot of those individuals at the end. And so here's our plan to identify, diagnose, and treat dementia. And so all of these things are happening right now in my lab and other labs at UNC. What is it about aging? And so aging is this real critical thing. And we need to understand aging in order to understand dementia because they go hand in hand. We now have new technologies to see into the brain with very high resolution. We can see signs of early disease, which is what we found in our recent study to identify these early clumps of tau. And now we're testing which risk factors actually promote disease. How do we slow this process down? Well, we've identified a handful of genes, and now we want to understand if we can get rid of them or express them in a adult brain, in this case, a mouse, but eventually in a human. Can we better diagnose dementia in early stages? And so to do this, obviously, you can imagine having access to patients and brain samples is critical. And that's why UNC is starting to bank brain tissue and store biofluids so that we can actually analyze biomarkers in patient samples. Now, what would be a good biomarker? Blood, plasma, urine, spinal fluid. Those are the easily accessible things from live patients that we want to test to see if they have early signs of this tau, this certain form of tau that we discovered. And so we want to combine that with MRI and PET imaging, which can be done in live patients. And to do this, clearly you can imagine we need to bring together lots and lots of people, both clinicians and researchers. And so I just want to close in these last few slides with this notion of resiliency. A lot of people think of something different when you hear the word resilient. Some people view the word resilient as, being tough or withstand some element or and so in the neuroscience field, the word resiliency means that you can withstand the burden of whatever forces may be driving your brain towards a disease state. Okay, and so I just want to give an update to my favorite uncle. This is Uncle Joe. He's actually my wife's uncle, and let's see, why is it not? Oh, there it is. Uncle Joe is turning 108 this year. He lives in Sarasota. His name is Joe Newman. He still drives. You can see his sports car there in the background. He has a fiance, and he has lived life to the fullest. Here's a picture of Joe's fiance, Anita. Anita just turned 100. So between Joe and Anita, they've got about 208 years behind them. And so this is a remarkable story. If anybody would like to read about Joe, he's in the news quite a lot these days. He's one of the oldest men in the United States, and he's showing no signs of slowing down. And I've spoken to him about what he attributes this longevity to. And, there's a lot of factors involved. There's genetics, there's environment, there's his positive attitude, there's his ability to stay calm and stress-free under very traumatic times. And he's had lots of trauma in his life. But at the end of the day, Joe has a quality that we call resiliency. And I'm sure if we peered deep into his brain, we would see his brain is very good at warding off the pathological signs of dementia. And how he did that is hard to know. But by studying people in this oldest old category, I hope we can start understanding the genes and the processes that contribute to resiliency. And we think that this gene we found, this HDAC gene, is one of the genes that is building resiliency. And I'd imagine if we could analyze the HDAC6 gene in Uncle Joe's brain, we'd find some pretty active protein that was doing a very good job even uh, into his upper 100. I mean, he's almost 108, and if you had a conversation with him, you'd think that he was maybe 40 or 50. So it's really remarkable. So I thought I would just introduce you all to Uncle Joe, and I thought I would stop here just by letting you all know that it takes an absolute village to do research. It's very difficult. It involves lots of coordination. It involves lots of egos, but it also involves lots of people with the right people that do this for the right reason. And I'm like thrilled and humbled to be a part of a group that all do this for the right reasons. And a lot of them are listed here, and you can just get an idea for how much is involved here. Dr. Gwen Garden is the chair of neurology, and she's orchestrated a lot of this. People that are well-versed in MRI, ALS. This is Dr. Dan Coffer, who passed away tragically earlier this year, who leaves a lasting legacy in FTD research. We miss him dearly, but he's left quite a legacy and big shoes to fill. And in addition to these really great folks, we have so many active collaborators that touch key areas of medicine, science, and all the things that you need to come together to develop cures, because you can't do this alone. So I acknowledge everyone. It takes funding, lots of funding from a variety of sources, and it takes a lot of hard work. And so I just want to end with this slide and say that please support this mission, because I think that we can probably make some great things happen. My contact information is there, and I'll just leave you with four bullet points here. Stay aware of the great things that we're doing at UNC, because I truly believe they're great. Be optimistic about what is happening and what will come, because it's been a long period of not many effective drugs, but I think great things are happening in the next 5-10 years. And I think we can make progress if we commit ourselves. And the reason I say that is because we've done this with HIV, AIDS, polio, and now, the most recent with COVID-19. I'm seeing that it's remarkable how quickly that happened. So when we want something bad enough, I think, we really can make it happen. And I would say that the 3 ways to make it happen are: first, you need money and resources. That's clear. But you also need creative, motivated people doing the work, and you need determination. I think without any one of these things, it doesn't happen. So we hope that we can put all these things together in the right pieces, and try to make some really good discoveries that lead to eventual therapies for these diseases. So I will stop there, and I think I'm sort of 4 or 5 minutes short of noon, so I think we can take a few questions.
Thank you, Todd. And yes, we're full of determination and a lot of want to fix this, right? So I do have a couple questions, and we'll try to get through as many as possible, but please know that if we don't answer your question today, you can just email it to me or to Todd personally, and we'll make sure it's taken care of and we'll answer it appropriately. But one question would be, is the memory lapse common to elderly people, forgetting names, etc., in the spectrum of the other disorders that you mentioned?
I'm sorry, could you say that again, Erin?
It's just the memory lapse that's common to elderly people, like forgetting names, etc. Is that in the spectrum of the disorders that you talked about today?
Yeah, and there's always going to be, with normal aging, there's always going to be this slow decline in memory. Now, there's a big difference between age-related memory decline that is pretty typical, and actually the brain shrinks a little bit as we age, versus pathological aging, which is when dementia sets in. Those are two very different things. We were not destined for a path towards Alzheimer's. There's this misconception that everyone goes on to get Alzheimer's disease or some form of dementia. It's not the case. You can age perfectly healthfully and gracefully, and if you suspect that something's not quite right, usually that's a hint that you should, talk to a neurologist. Usually, when people have some sort of inkling that something's not quite right, that's usually a good sign that you should go do a consult at least with a neurologist.
Thank you. Do plaques and then tangles always cause dementia?
That is actually a very good question because um, in every case of Alzheimer's disease, you have plaques and tangles, but plaques and tangles don't always cause dementia. Hopefully that makes sense. I'll say that again because it's such a good question. In every case of Alzheimer's disease, you have plaques and tangles, meaning you can't have the disease without them. But you can have plaques and tangles and have resiliency to them and not show signs of Alzheimer's disease. Hopefully that made sense. A very good question, which we still don't understand.
That's fascinating. Assuming that you have some signs of dementia, such as forgetting where you leave things or accessing words that you know, does that mean you will move to full-blown Alzheimer's?
Not the case. Everyone's progression is very different. You can not progress at all. You can progress within a year or two. The differences from person to person are so striking. There's no rhyme or reason between the length of time it takes to progress. It could involve genetics. It could involve risk factors. It's so dependent on that person and that brain and other socioeconomic, potentially education levels, all these other, ethnicity, all these other things factor into it. And it's just a difficult question. I can't say.
Well, you tried, so thank you.
Yeah.
One will be, will the gene therapy possibly restore the hippocampus?
Yes, I'm very hopeful. I think that what we're testing now is exactly that question. And so whoever's question that was, uh, they it, we'll keep you posted because we're going to see if this gene therapy can restore the hippocampus size and also prevent the deterioration that occurs there in the mice once we've given them this virus. So we have the virus now, and we will keep you posted. Good question.
I've been told that there are no tests to detect a very early Parkinson's. Is that true?
That is true to the best of my knowledge. There's a big push to identify biomarkers for Parkinson's, and people are looking in all places, blood, plasma, and I have a colleague at Duke who's looking in urine. Um, and so early biomarkers for any neurodegenerative disease are essentially unknown. That includes Parkinson's and Alzheimer's disease. And we think what we've found, this early form of tau, may be one that gets us to an earlier stage, this midlife time frame, which I talked about. So we're hopeful, but none exist currently.
Thanks. If a parent has dementia, are you at higher risk to develop dementia?
Not necessarily. Now, you could be tested for the Alzheimer's panel. And if you wanted to know if you were an ApoE carrier, for example, your risk of dementia goes up if you have certain at-risk genes. And you could know that information if you were so inclined. I guess I would pose to that person whether they, and it looks like Janice, if you were so inclined to want to know that information, you could. And if you, I think it's more of an ethical question of whether you want to know that or not. But just because a parent has it, keep in mind, Alzheimer's is very prevalent. So just because a parent has it does not necessarily mean it's a genetic thing. Most cases of Alzheimer's disease are sporadic, which means they evolve randomly from person to person.
Thanks. Do you have any evidence that ECT treatments for depression could result in sufficient brain damage to result in Alzheimer's years later?
And that looks like from John. I don't know the answer to that question. There is some evidence that trauma in any form can head trauma can result in dementia, but it's very particular to the type of trauma. There's also some connections between post-traumatic stress and other types of psychological trauma that might be related to Alzheimer's disease. My personal view is that you have a bucket, a stress bucket, and once that bucket is overflowing, and a lot of things can go into that bucket, once that bucket is overflowing, it could put you at risk. Now, what those individual things are and how much they contribute to your bucket is very is debatable. And so I don't exactly know the answer, but all I can say is that trauma and stress are implicated in progression, but it's not really proven.
Thanks.